box, the type of cell produced. In mitosis, it's a somatic cell. In meiosis, it's a gamete. Now, perhaps you didn't say those exact words, but you said something that means that. For example, for mitosis, if you said something that's not a gamete, you are correct. It produces everything but those. Meiosis, if you said a sperm or an egg, something like that. Even if you said diploid and haploid, I would accept that. Those would all be good ways of describing it. Or if you said germinal cells. Now, germinal cells are the ones that do meiosis, but not the ones that are produced. So for the moment, we'll accept it, but there's a small little distinction, and we're going to actually go over 2 examples of meiosis today that will clear that up for you. Do homologous chromosomes align? This is more or less a yes or no question. In meiosis, yes. Specifically, meiosis 1. In meiosis 1, homologous chromosomes line up next to each other. What we're really asking here is, does a tetrad form? And so the answer is no in mitosis. The location, so where is this happening? There's a lot of ways you could describe this. I would say here is where I would put germinal cell, and here is where I would put any other body part. So you could repeat the word somatic there if you wanted to. If you were more specific and you said something like in an ovary or in a testicle, that would be acceptable because that is a location that they are happening in. Number of divisions, in mitosis there's 1, in meiosis there are 2, because there's meiosis 1 and meiosis 2. Is there replication of DNA? Yes and yes. Specifically before meiosis 1, but of course there's replication of DNA. It happens in interphase for both of them. How many daughter cells are produced? 2 and 4. What is the ploidy of cells? Now generally it's diploid and haploid. Of course, if the cell that started mitosis was polyploid, then it would be the same ploidy, but this would be your go-to answer. Do the daughter cells divide again? They absolutely can here, and they do not in meiosis because their cells do not go into interphase again after that. And finally, is the genetic material identical? Yes for mitosis, no for meiosis. So there's 9 rows, there's 2 boxes on each row, so count each one as a 0.5. And give yourself a total out of 9, please. So the first one we'll talk about is spermatogenesis, and it kind of has 2 parts to the word. Does anyone know what the piece of word Genesis means? Maybe you know that Genesis is the first part of the Bible. We're at a Catholic school, maybe you know that. You know what it means? It means the beginning. So spermatogenesis means the beginning of sperm. Where do sperm come from? So spermatogenesis is the way that sperm are made, the way that they are produced. There are 4 words that we're going to describe or define when we talk about spermatogenesis. And each of them is represented on this picture. It's also represented on the picture in your textbook. During spermatogenesis, we start with a spermatogonium. This is a germinal cell. Now I'm going to mention something about when this happens in a person's life because it's different between a male and a female. Spermatogonia, which is the plural of spermatogonium, are present in the embryo and at birth. And they are what a male has until puberty arrives and something else is going to happen. So in the embryo and at birth, until we get to puberty, spermatogonia, which is the plural of spermatogonium, is the type of cell that exists. A spermatogonium will undergo mitosis. Mitosis. Yep, all three of us. Now the spermatogonium undergoes mitosis. So you'll see right here on the picture, it says mitosis. The reason it undergoes mitosis is because we need to make sure that there's a constant population of cells that could eventually produce sperm. So this cell over here, this daughter cell, repopulates the spermatogonia. Make sure that we always have spermatogonia present. The other daughter cell from mitosis will continue on and become what is called a spermatocyte. We can say first order or we can say primary. It would be the same thing. So the spermatogonium will eventually become the spermatocyte. Spermatogonium is just undergoing mitosis, so nothing fancy is happening with its number of chromosomes. A spermatogonium would be diploid. Now after that, we have our primary or first order spermatocyte. This primary or first order spermatocyte would also be diploid, right? It hasn't undergone meiosis yet. This cell is going to undergo meiosis 1. And this part of spermatogenesis occurs once we have puberty happening. So it's not until puberty hits that this part of dividing is actually occurring. Now, after we have our first order or primary spermatocyte, it becomes second order or secondary spermatocytes. These are N, so these are haploid. So at the end of meiosis 1, we have reduced the number of chromosomes. Spermatocytes, again, this part only happens during puberty, and these cells undergo meiosis 2. Now at the end of meiosis 2, those spermatocytes become spermatids. Spermatids are again haploid, so they have N chromosomes. Spermatids are not mature sperm. Mature sperm require a little bit more time to develop. So spermatids we will call immature sperm. And at this point in the course, I don't need for you to know specifically how they get from spermatid to mature sperm. That'll be for another chapter. But just notice the difference. A spermatid still looks round, whereas an actual sperm cell is long. It has a distinct head, middle, tail. And that's required for fertilization to actually take place. So this is what happens during spermatogenesis. We start with the spermatogonium. It undergoes mitosis to become a spermatocyte of first order. Those two cells are both diploid. Once the spermatocyte, the primary spermatocyte, undergoes meiosis 1, now we have haploid cells for the rest of it. Now, does anyone want to ask any questions about sperm? I want you to notice very specifically that there are 4. And I'm pointing this out because when we go on to the next slide, where we're doing oogenesis, which is how eggs are produced, there will not be 4. So again, if you need a reference, page 569 of your textbook has a picture that has both sperm and egg production. You could also go online and print out this or the entire package of notes if you wanted to. On the next slide, we're going to talk about how eggs are produced, and it has a similar type of word beginning the process. The word has genesis in it. Here it's called oogenesis because usually the words have an O in them. So oogenesis is the beginning of the egg or of ovules. And there's a very key thing that is happening in oogenesis. The key thing that is happening is asymmetrical. So if I look at the word, it has an A at the beginning, which means not scientifically. So it is not symmetrical. So asymmetrical cytokinesis means that when cells divide, we're not getting the same amount in both of them. So we start with an oogonium, just like we started with a spermatogonium. This is our germinal cell. Like you can see in the picture, this is diploid. It has 2 N chromosomes. And just like the germinal cell in spermatogenesis, it undergoes mitosis. Now, notice the line on the picture. All of the stuff that is above the red line happens before the female is even born. So she is an embryo. She has not been born. She is still inside of her mother's body, and this stuff is all happening already. After this mitosis, we wind up eventually with our primary oocyte. So your primary oocyte, just like your primary spermatocyte, is the one that is undergoing meiosis 1. Now it doesn't finish meiosis 1. You'll see that it arrests, which means it stops in prophase 1. So it doesn't finish meiosis 1 until puberty occurs. And then there's a hormonal signal telling the woman's body or the female's body to continue on with the production. This means that a female is born with all of her eggs. She is born with a set number, and that is the number that she is going to have. So if we were to compare a male and a female, the average, and it's really just an average because it's not identical for everybody, but somewhere in the neighborhood of 400 is how many eggs a female is typically born with the capacity to produce. Males, on the other hand, could produce sperm basically indefinitely and in the billions of quantities. Eggs have a much more limited number, and you'll see it takes a little bit more to get one egg produced than it does one sperm. So once puberty hits, my primary oocyte will continue meiosis 1. So I'm just going to add a note here. This is meiosis 1 that we're finishing. Then we end up with a secondary oocyte. The secondary oocyte is haploid. And I want you to notice that I am not getting 2 daughter cells. Normally, after meiosis 1, I produce 2 cells. But instead of producing 2 cells, I am producing a cell, and I am producing what is called a polar body. The polar body is not going to become an egg. This is an example of asymmetrical cytokinesis. The secondary oocyte is bigger. The polar body is much smaller. The polar body is haploid. It has half of the chromosomes. The secondary oocyte is haploid. It has half of the chromosomes. But the polar body gets much less cytoplasm. It gets less nutritional elements. It gets less of the organelles. So aside from the DNA that's being split apart, it's not really getting any of the rest of the cell. That primary oocyte is going to divide and make one really big cell that has most of the cytoplasm and all of the nutritional elements, and one really tiny polar body that basically just has half the chromosomes and is not going to continue on and result in reproduction. Now the secondary oocyte gets paused as well. So the secondary oocyte is going to undergo meiosis II, but it's going to stop in meiosis II, specifically at metaphase, until ovulation and sperm entry have occurred. So if a female produces a secondary oocyte, it will not finish meiosis II unless it gets fertilized. So fertilization is necessary to complete meiosis II. So that secondary oocyte will not finish meiosis II unless first a sperm comes along and fertilizes it. That means that at the end of this process, I have a fertilized egg, which is diploid, and a polar body again, which is haploid. So at the end of spermatogenesis, I have sperm that are all haploid. If oogenesis actually completes, I have my little ovule here that was haploid, but it will be diploid at the end because it had to be fertilized for this process to be finished. It's weird. Now, why does this have to happen, specifically this asymmetrical cytokinesis? This is because the egg provides everything to the embryo. The only thing that the sperm provides is some DNA. It does not provide nutrition. It does not provide energy. Like usual, the girl's doing everything. The guy's doing nothing. I'm joking. That's me being sexist. But the egg has to provide everything that the cell needs except for that little piece of DNA. So except for the haploid number of chromosomes, the sperm is not providing any other cell structures to the embryo. Now, does anyone want to ask any questions about oogenesis? It's definitely the trickier of the two. Spermatogenesis resembles meiosis, sort of phase for phase. But oogenesis is weird because it stops at different phases. You finish with something that's diploid. All right, so we have one more thing to go over, and it would be the idea of twins. So if you did the suggested reading that I suggested yesterday, you would have read about formation of gametes, so spermatogenesis and oogenesis, and then you would have read a little bit about twins. Before I go there, does anyone want to ask anything or maybe clarify anything that's up on the screen? Yes. So if I was thinking about meiosis in general, what happens in meiosis? I start with one cell, I end up with two, and each of those cells divides and makes two more. That is your typical meiosis diagram. What we're saying is that in oogenesis, at the end of meiosis one of these things is not going to be a full large cell. It's going to be small. So it's not even going to divide again. It doesn't continue on. This would be your oocyte, the one that's going to continue on. And of these two, one of them is not going to have enough material to be an actual gamete. So what we're saying is each time the cell divides, one of the two daughter cells is bigger than the other, and only the bigger one continues on. So if I'm looking at this picture here, this would be my fertilized egg. This would be my secondary oocyte, and this would be my primary oocyte. So we're undergoing meiosis, but we're only getting half of the daughter cells each time because one of them is a polar body, meaning it's small and it has nothing to support itself with. Does that answer your question? Perfect. So our very last thing is about twins. There are two types of twins that exist: dizygotic and monozygotic. Di means two, mono means one. So a dizygotic pair of twins are twins that come from two sperm, two eggs, thus two zygotes. So dizygotic twins are in the very rare case where a woman has released two eggs. There are always more than two sperm. If there were only two sperm, it would be a crazy, ridiculous scenario. There are hundreds of thousands of sperm at a time. But the woman would have to have released two eggs at the same time. If two sperm fertilize two separate eggs, then you can have two separate zygotes. Dizygotic twins are what we would call fraternal. They are no more similar than any regular brother and sister. Fraternal twins could be two females, it could be two males, or it could be a female and a male. And that's because there's nothing necessarily identical about them. It was two different sperm, two different eggs. So very frequently, fraternal twins do not look a lot like each other, no more than, say, any of you look like your brother and sister. I don't look like any of my brothers and sisters, so I can't use this on myself, but I'm sure some of you bear some sort of resemblance to your siblings, and that's the similarity that you would find in dizygotic twins. Monozygotic twins are the ones that we would call identical. They have to be the same gender because they have identical DNA. Monozygotic twins happen when one sperm and one egg make one zygote, but that zygote develops into two groups of cells. Since they originated from the same sperm and the same egg, they all have the same combination of genes, so they should have identical characteristics. So I have a little picture here to represent what is going on. The one on the left is monozygotic. The one on the right is dizygotic. So on the left, you see an egg. She's waving and saying hello. This was a French picture, so she's saying cuckoo. And the sperm is like, oh, yay, cool, there's an egg. The sperm fertilizes the egg. Cells start developing. And then right here, the group of cells divides into two. That means that two separate embryos will develop, and you will get two people, but they started with the same genetic information. In dizygotic twins, notice how there was a separation the entire time. There were two eggs, there were two sperm, there were always two groups of cells, so they were never part of the same group of cells.